For a long time, humans have tried to use electricity to probe biologic cells and tissues. Way back in the 1700s, Luigi Galvani was experimenting with electricity on the legs of dead frogs to gain insights to the electrochemical aspects of biology. In modern times, we still use electric properties of certain cells to monitor and gain insights on the human body. Tools such as electrocardiograms, or EKGs, and electrodes placed on someone's head to read brain waves are ways that electricity can be used to monitor the behavior of cells and organs. Both of these techniques provide continuous monitoring of living tissue in a way that is not invasive, which means it doesn't affect the way the tissue functions. While these are both valuable tools, the cells and organs they can monitor are limited to ones that are electrically active. In the early 1980s, two scientists working at the General Electric Research Facility in New York State set out to use electricity to monitor cells that themselves were not electrically active. Their idea was to apply an alternating current, the same kind of electricity that flows in the power lines, across a single cell layer to monitor the activity of the cells. By keeping the magnitude of the current low, this technique would not affect how the cell layer behaved. In 1984, they published their experimental setup for this technique and coined the term electric cell substrate impedance sensing, or ECIS. Impedance refers to the difficulty of a current passing through a circuit. The way it works is by applying a small alternating current over a single layer of cells growing on one of the electrodes and monitoring the voltage. As the cell layer grows and develops, the measured voltage changes, and by knowing both the current and the voltage, the impedance can be found by Ohm's law. The voltmeter is hooked into a computer, which can record the data over long periods of time. Now, where this technique gets interesting is cells actually behave differently when exposed to high frequency versus low frequency current. At high frequencies, the cell layer acts like a capacitor, where the charges can build up on either side of the layer as the current direction changes. At low frequencies, however, the cell layer acts like a resistor, where the electrical charges have to pass through the spaces between the cells. This means high-frequency measurements can be used to determine how much of the electrode area is covered by cells, while low-frequency measurements can probe something known as the barrier function, or the cell's ability to resist electricity. This method of measuring allows scientists to test a large number of different coatings and chemicals on the cell's ability to not only initially stick to a surface, but also to form the barrier function. This is important when it comes to designing different implantable devices like pacemakers and artificial joints. It's also useful for monitoring how the cell layer can heal after it's been damaged. Some ECIS devices are even capable of electrically wounding the cell layer by passing a dangerous amount of current through the electrode, and then they're able to monitor how the surrounding cells move to repair the damage. So just like other electrical methods, ECIS is able to continually monitor a cell layer without interfering with the function of the cells, with the added benefit that the cells being monitored don't need to be electrically active. It also eliminates the need for cells to have special stains applied to them to be monitored by optical means, thus reducing the cost of experiments. All of this makes ECIS a simple yet powerful technique for cell layer monitoring.